So I do research up here in Yosemite about bacterial endophytes, which live inside of conifers. You may be wondering, what in the world is an endophyte? Well, if you break down the name, endo, inside, phyte, plant. So they're bacteria or fungi that live inside plants. Now, the interesting thing about them, why study bacteria and fungi living inside plants? Well, it's because of the beneficial properties they can provide to the plants they live inside, such as, which I'm most interested, growth promotion, but also in addition, protection from disease, insect pests, and also drought. All right, so once I'm safely in my sterile equipment, then I can choose buds and needles to sample. So what we're going for is healthy tissue because endophytes don't cause disease. And I'll sample just a very small portion of the needles and buds because I'm interested, are there different bacteria in the needles as compared to the bud? I can study how does the relationship between endophytes and conifers develop over time. For example, an established tree like this one may need more bacteria to help it survive drought and disease, whereas a young tree, who knows, maybe they need more growth promoters. But I guess I'll find that out as the study progresses. One of the main reasons I chose Tuolumne Meadows as my study site is the amazing proximity to UC Merced. Also, there's a great cooperation between UC Merced and the Natural Reserve System that allows me to have access to this wonderful research site. I also have use of the Sierra Nevada Research Institute's field station if I need to camp over, and I have other study sites in the area, so that helps me have access to those. But mostly I get to backpack, which is awesome. Once I have sterile samples, then I can take them home to the lab. So we start off with crushed up plant material pressed onto an agar surface. As I use different amounts of crushed up material and liquid, I'll slowly begin to see bacteria growing on the surface, like this colony. Then I put the plate in Bardot. This is Bardot, a rapid analysis method for identifying bacteria just by shining a laser through a colony. The stage that the plate is sitting on moves along the shortest route from colony to colony and shines the laser through. There's a little camera underneath that collects the scatter pattern of the light as it bounces through the bacterial colony, which allows us to identify bacteria because each of their scatter patterns is unique and different based on how they grow on the agar surface. So Bardot is an amazing tool to rapidly identify bacteria instead of waiting months for sequencing results. Before I used this technique for endophytes, it was used strictly for identifying food-associated pathogens. I said, why couldn't I use it for identifying bacteria that I grow out of trees quickly? So it's great for my project because I'm trying to figure out all the endophytes that I can grow from Tuolumne Meadows, which are hundreds and hundreds and hundreds. The field of microbial ecology has been developing to understand the big picture. We are really interested in the human gut microbiome because we know it's important to our survival. So there's a similar interest in the bacterial endophytes within conifers because they might be just as important to that conifer in harsh circumstances.